with all that, we've got PowerPoint being captured. So let's go ahead and go through. This will probably follow the same pattern as last time, where I'll be able to get through probably parts 4 and 5 today, and I made those a little bit more interactive. Part 6 is less interactive, and it's just some examples. But as I was doing part 6, I was like, you know, I think I want to not alter the last assignment, but expand it. So if you've already found a good paper for predictability of evolution, great, go for it. If you haven't, you're going to have another option for a topic that you can choose a paper on. Basically, you can choose a paper on cross-reactivity in the immune system. And that's what part 6 is all about. So when we get to that, I'll explain a little bit more. Basically, it's because I have to sit here and listen to you. I want you to teach me. And so I want you to teach me about evolution and predictability if you want. Or if you want to teach me about cross-reactivity in the immune system, you can also do that. So both of those are complete open options. It's already even posted online. Because I like this stuff. I like thinking about why things are cross-reactive. And the one question we want to answer is, as a person who studied the T-cell branch of the immune system, when I heard about variants coming out and escaping the immune system, I was worried, but not as worried as some other people seem to be. Because T cells are less responsive, they're less likely to be hurt by mutations going on that would hurt antibodies. Now, why is that true, and what does it mean? It doesn't mean that T cells are going to save us from ever getting sick again. But it does mean they have a certain advantage that actually has some advantages over what antibodies have. Different structures, and there's some similarities even at the structural level. We'll talk about those. Before I do that, I actually want to go back and I want to find out that I tell you that I got this advertisement. You get also this scientific spam, and by the way, if maybe you've gotten something like asking for your contribution to a special issue of some weird journal that doesn't even have its name spelled right or something like that. Just ignore that. That's just as much spam as the weird calls that you get these days. But there are companies trying to sell products, including companies trying to sell ELISA. And you see right here, they even have a little workflow of ELISA. I'm giving this to you not so much because I'm going to ask you to run an ELISA, but you should understand when you see these things, these are the actual steps. If you haven't had the chance to do ELISA, say in cell biology or something like that, you at least can understand the steps, and maybe someday you'll get a technician job somewhere, and you'll be asked to run an ELISA, and you'll be able to say, oh, I have seen that before. There it is. And they might buy these things, and you might eventually actually even talk to someone about which one of these should I buy, and you talk to the companies. So this is actually a business area. So all that's to say, ELISA is good for measuring antibodies, because it's good for measuring protein-protein interactions. But the immune system is more than antibodies. So I just want to review a little bit for parts 1 to 3. Just tell me something that was, what did the antibot do? It's a very vague question. But if I had to ask you to say in a sentence, what did the antibot do? There's several ways to answer that. How would you answer that? Okay, so you're talking about you're kind of talking about the front end of what it does. The fact that there's randomization going on and it's testing a lot of different possibilities. Perfectly valid answer to that. There are other answers as well that can be just as specific, that can be down the road. I don't know if you remember what you saw the robot actually doing. We didn't say that's what the big picture purpose is. We can also drill down. You can answer this question another way. Drill down to say, what did it actually do physically? What did you see it doing when you saw the Everything is Awesome video, if you had the chance to see that? Right. So you saw the ELISA plates, and you saw it actually moving those in and out, and it was like pipetting in and out of those. So it used ELISA to measure the randomness somehow. And I'm still deliberately leaving out more parts to see if someone wants to pick up. What else did it do? Yeah. So surface, and that's how they were using it. That's specifically how Wells was using it. He was starting with a protein. What's that? Yeah, you are actually answering the second question. as well, because you're answering why. They didn't actually grow any tumor cells inside it. Oh, well, they might have had tumor cells at one point. But they were looking at protein-protein interactions. It didn't necessarily even need to have the cells. But the whole reason why they were looking at it is because at some point they had determined cancer cells have this protein on it. So we want to try to be able to grab onto that protein, to detect it, to even deliver a toxin to the cells that have that on their surface. So, yeah, the main thing about this is they went through all that trouble to find a cell surface protein, because antibodies cannot enter cells. Cells have a big barrier to them, and you can't go inside a cell. It's the whole point of having a cell is to have the cell membrane preventing stuff from getting out and stuff from getting in. The problem is things do get in. Viruses infect cells. Then you have foreign material inside the cell, hijacking the cell for its own purposes. How can your immune system detect those things? So antibodies, of course, they're faster, they're easier to measure, they're just proteins. To work with other branches of the immunity, you have to work with cellular immunity, which means you have to work with whole cells and culture them. I have a little bit of experience with doing whole cells, but actually usually what I would do is I would take a step back again, and I would look at the proteins that mediate cellular immunity, because honestly, I don't like working with cells. I like working with proteins. They're much more forgiving. But it's a different branch of immunity. It can do different things, and it's trickier to deal with in the lab. It's also slower in real life. So the antibodies are fast and easier to deal with. But there's a whole other branch, and this branch is important. This is interesting. This is a letter that was just recently sent to the FDA. There are 60 signatories on it, most of them prominent scientists running their own labs. And they were advocating that FDA look at T-cell responses to COVID rather than just antibodies. Remember, if you've looked at any of the data that comes out, you always get all these things about antibody binding, you get things about prevention of infection, hospitalization, the clinical endpoints, and then you have antibody binding on the other hand. But what about the T cells? There's this whole other branch, and again, it's hard to look at. That's why they don't look at it. But it's important because if you don't account for it, you might be a little more worried than you should be. You might be only thinking about half of your immune system. And so to get a realistic picture of what's going on, you need to know what's going on. And by the way, you can have autoimmune diseases that are T-cell mediated as well. So as with anything with immunity, it's a double-edged sword. We need to understand it. So I want you to think back to summer 2020. And do you remember anything about what it felt like in summer 2020? Maybe, do you remember what you had heard about vaccines? It's actually a little bit hard now, but now that we know that something's gone on and we have, SPUs required everybody who can to get vaccinated and things like that. Do you remember anything about what you thought would be coming down the line as far as the vaccines or things like that? Or what you thought about COVID as far as what would happen? It's a deliberately vague question. But I want you, do you remember anything about what it felt like in summer 2020 when you were thinking about COVID, antibodies, vaccines, and stuff like that? Eric? That was the first glimmers that it might actually be ready that early. I remember saying that it would take 15 to 18 months from April, but it took 9 months, which is good. Vaughn, what do you remember? Right. Yeah, there's a question about the seasonality. I actually have a paper on that, by the way, if you're interested, that suggests that seasonality is definitely real, but we don't really know exactly. It's hard to tie down exactly what happens. Every virus is a little different. So what would the changing seasons do to the seasonality of this relative to other coronaviruses, relative to this being new and more infectious and a worse disease than the common cold, for example? Yeah, so the questions about seasonality, and the paper that I have, I think it has a very suggestive thing. When you think about seasonality, you think about the virus getting worse or better with the changing seasons. This paper says, what if it's the immune system itself that defends you worse or better with the changing seasons? What if we're looking at the wrong thing and thinking about it all as being about the virus? Very provocative. If anybody wants to dive into that, I've got the paper. But that's a good question. That's a huge question. I actually had a graph of what the other coronaviruses did. And I remember in fall 2020, I deliberately rearranged our entire lab section so that we would have the computational labs last in Biochem 1, because I was sure that there was likely to be another wave in December from the seasonality of it. That worked out. It's actually slightly later than I was worried about, but there was a wave that winter, obviously. So yeah, that's one of the things. Yeah, think about any things like that. Any other stories about, like, you remember what it was like, of what you were wondering about?
I remember hearing about Project Light Speed, realizing
yeah, warp speed, I think, actually. Is that right? Yeah. Anyways, yeah.
Right. Anyway, I just remember the gears have really started turning, and the government always was just follow.
There's an emphasis on that. And then one of the two vaccines that came out actually didn't get that much help from Operation Warp Speed, but the other one did. So we still have to completely add up exactly how much it did, but at least there was a lot of money going into vaccines, and it appears to have paid off because it wasn't 18 months, it was 9. And so this is from September 2020, and I remember this headline came out because we didn't know how long would your immune system remember once you had been attacked. And they had this headline which really bugs me. People are protected up to 3 months. If you remember, we didn't know if we would have durable, lasting immunity. And of course, people were always putting it together. They're putting immunity to catching it, to immunity to the hospitalized with it, to dying from it. 3 different levels of immunity, but they were always thrown together. So this data actually was wrong. Not wrong, it's not a lie, but the word should be for at least 3 months, because they measured 3 months of protection. That's all they had to measure at the time for the study. That's, and then they have this may fade in as little as 2 months. That's an extremely pessimistic view of the data. The data were showing that you had strong antibody response for 3 months, basically. But you see how that got translated. So we've got to remember the immune system has multiple areas. In fact, 3 of the branches, we've talked about one of them. We've talked about antibodies, which are made by B cells. But then you've got 2 other branches other than antibodies. You've got innate immune cells and whatever antibodies are actually there, and it's normal for antibodies to fade over the months after infection. This is something that shows a coronavirus infection, and it shows that the antibodies go up after infection, and then by 12 weeks after inoculation, you see they're dropping because the virus is not around anymore. Now, I said coronavirus, and you might have interpreted that as COVID, unless you read the fine print here. It's actually one of the common cold coronaviruses, the 229E coronavirus, which is 60% similar to the COVID. And so you see that it's normal for this to fade, and yet you still remember, despite your antibody serum levels dropping, there's lots of other ways for your immune system to remember. And so this was something that came out in August 14. This was the first good study that I saw that really saw that you had persistent protection. And so you have, and they actually measured not only what were the antibody levels up to 3 months after infection, what were the memory B and memory T cell levels. And the important thing about this is they detected every level of protection. They detected not just the antibodies in your serum, but that you had cells, both of B and T variety, that take longer to develop, but they last for months or years. And we've been able to follow this up. The general idea that we do have lasting immunity, maybe not immunity to infection, because the thing is so infectious that it's hard to completely have a shield that will put up everything against it, but definitely immunity against severity. So this is where they were looking at COVID recovered subjects, and they were saying You might only need a single vaccine dose to achieve the peak antibody and memory B cell responses. That's not to say that you don't need a booster. It's just to say that the original dose works well. And of course, the problem that this doesn't mention is the virus changes. So as the virus changes, we have to keep up with it. All that is to say, the vaccine, I did not know in summer 2020 how good the vaccine would be. And honestly, if you ask me what a percentage would be that I would want, I would hope for about 60, 70% effective, maybe 80% effective. And they came in, and Francis Collins talked about this for those of you who watched his talk, came in, it was 95. If you're expecting to get 80% on a test and you get a 95, you're pretty happy, right? And that's pretty much the equivalent of this. But there's other tests coming. You can't just sit back. And so it's good, but it's not the end of the story, right? So we have this, and this is the first time that we were measuring it. Of course, in the time since, we've run lots of studies. I have one of the latest ones coming up in just a couple slides. Now, the other thing about the immune system is that when you catch it, and people in my family have actually gotten it, and I've noticed that my kids, when they got it, before they were vaccinated, they actually had similar response to me when I had the vaccination. There's these chills that you get sometimes. And the chills are actually your body putting energy, it's running metabolism. So as people who've been through Biochem 2, you can understand a little more about this, that you're putting a lot of sugar into those side effects. And one of the reasons why you feel fatigued is you are doing a lot of cellular replication. Your immune cells are expanding. So, and side effects are actually connected to the fact that if you had worse side effects, at least there's a silver lining, you made slightly more antibodies. But even the people, and this is on average, some people without side effects made a lot of antibodies, vice versa. Everyone's different, right? But if you had fever and chills, at least you could tell yourself, that's your immune system making itself. That's your shield forming around you. And they also found out that everyone made good memory cells. The side effects were for like the antibodies in your blood at the time. But everyone had good memory cells, and even those without side effects, they would tend to have fewer antibodies in their serum, but they had the book to make more antibodies stored in the memory cells. So this is actually, and this is something, I don't want to have confidentiality, so I'm not asking you to identify anyone. But just like I'm asking for stories about the other ones, I'm wondering if you ever noticed if somebody that two people you know are related, mother and son and things like that. Is there a possibility that there could be a genetic component to whether you get severe side effects or not? Do you have any observations about that? What do you think?
Pretty sad story about it. So when I was graduating, there was a patient who came in who was North American, and he was only 35, had a pretty fast birth, and 2 weeks later his younger sister.
Oh my, yeah.
So they went through that thing when 2 people have birth, just really unexpected.
Yeah, statistically speaking.
Just brilliant to
Right.
unlikely unless there's some sort of duplication.
Right. So that's even. I was just sort of thinking that we would have a knowledge base that would include side effects, but that's actually clinical outcomes. So you can see that there's the chances of that happening to 2 random people very low, but there's something going on, and I have hope with looking at the immune system, we can figure out what is the factor that they had in common. And then we could come up with a treatment when we see people with it, and we would also know that they were at a very high risk if we knew somebody had that gene or whatever it was. So yeah, some of the genes we're going to talk about today could be in that area. Yeah, any other thoughts about that? Yeah.
I worked at birth center, and one of the moms had COVID.
Mm.
She COVID, and they were breastfeeding, and they had that baby's antibodies
Yeah.
for COVID. And that was real surprise, but she also had, the baby had antibodies for COVID. And so she actually, the baby had more than the mom.
Okay. Yeah, and that can happen with iron at least, that the baby ends up with more iron than the mom is anemic. So I guess it makes sense that it would happen with antibodies as well. Sometimes the mother's body is a little bit too giving. But at the same time, that's more protection for the baby. So the mother probably is glad that that happened. We even know the molecular mechanism for that. And we could detect if somebody has like a mutation in that gene maybe, and we would know that they wouldn't be able to pass the antibodies on to their child, and we'd know to have some other course of action in that case. So yeah, there's definitely all these connections between genes and immunity. The one thing I was just noticing, my wife and I had very different patterns of reaction to the side effects. And so the thing about side effects is you feel sick if it hits you. You don't really know, and that's one of the things that actually affects vaccine uptake. I would like to be able to predict more so we could say to someone, oh, you're probably going to have to take work off tomorrow after you get your vaccine. I think we underestimate how hard that is for some people. And we wonder why, we're like, why didn't they take the vaccine? They must be anti-vax, you know. Maybe they have to eat, you know, and they can't afford to miss a day, things like that. So there's a lot of other factors, ways we can support people and get people to do this. And if there is actually an MHC molecule that's correlated with severe side effects from the second dose of Pfizer. And actually that was one person in my family that had that and not me. And so I would love to test our genes and to test this hypothesis that this other person had this form of MHC and I had a different one. I did not. And that could explain it. There's a ton of other explanations, right? But we are beginning to be able to do some things like this. So this is very true, and the immune system... Oh, yeah, Alessandra. Yeah.
So we talked from one about kind of the process of how the mRNA vaccine works.
Right.
But here you mentioned the MHC, right? Which
Mm-hmm.
is specifically for T cells.
Right.
That's the adaptive immune system. And so I'm wondering, why do we get the side effects so early on after vaccination if the adaptive immune system is seen to take a couple of days
Right.
to actually do?
That's because those side effects, I believe, are B cell, and they're already present B cells that can react already. If you think of the clonal expansion, you know, out of those millions of cells, you have like 3 or 4 that actually can react. And I believe that it's connected to those. In fact, I do have some kind of paper that directly connects side effects to memory B cell formation. Okay. But we don't really know for sure. I have one paper, right? You know, there's a lot of papers that it takes to do that. But that's what's reasonable. It's not reasonable for someone to say, oh, it has to do with T cells. Because T cells take like a week to really kick in even. So, yeah, at best. Yeah, and there's all sorts of different types of B cells and T cells. So, for example, there are some side effects to some flu shots that have been traced to transitional B cells. Now, we are only going to be talking about like the classical types of cells, and immunologists can get really down into the different types of cells. How do you even define a different cell type? And here's one that was associated with an overreaction to a flu shot. And this predates COVID, if you'll notice. The other thing is, if you know how COVID proceeds, one of the ways that COVID kills, it has to do with the overproduction of IL-6, which is an interleukin, a small protein. And in fact, there are some companies that even do interleukin therapies, like they make it up just like you make up an antibody. And so the protein production that you did in lab, if you took the one with me, was actually similar to what they do. And so we can manipulate these. If we can manipulate the level of the protein in the body, here's some idea. Maybe we can, if we have this IL-6 that's causing the cytokine storm in the person's system, maybe we can stop it not by killing the virus, but by turning off the body's immune response that's overreacting, that's more harm than good. And they literally have an idea where you take your blood and you filter out the inflammatory cytokines, filter out the IL-6, run it through a column, and bind up the IL-6, and then take the stuff at the end and give it back to the patient. If you think about this, there's all sorts of wrinkles to this, but it is a possibility. So the one thing that makes immunity so confusing is not just all the different types of cells, but all the different types of responses. And if you think about it, if you talk about your immune system, you can think about allergy and autoimmune diseases as well. The immune system is supposed to respond to infectious agents, but is not supposed to respond to innocuous substances. So when you have the white boxes, you have the immune system choosing the right road. But in every single one of these, we sort of want it to choose differently. And the fact that it is able to choose enough so that we can function is actually pretty amazing if you think about it. It usually is pretty good at activating against infectious agents and not activating against innocuous substances. When we graft an organ in, we want it to not respond to that organ. We also want it not to respond to our own organs. Therefore, if you graft an organ in, make it more like the recipient's organs as possible. Make it as much similar to them as possible. That's why relatives are good organ donors, because they have similar molecules on the surface. But of course, tumors are the other thing. They're your own cells, but they've been transformed into this malignant zombie-like mode. And we want the immune system to respond to them. And there's a lot of the immune response that actually can and does respond to it. It's why I did some of this immune research at the Fred Hutchinson Cancer Research Center. Immunity can protect you from cancer. It probably does, and you don't even know it. So that's why I like this. It has this whole yin-yang balance of things where you're trying to interact. You need to interact with the outside world. You need to maintain the integrity. So there's interacting with outside. There's maintaining the inner integrity of the body. If your immune system reacts too much, you get tissue damage. If it doesn't react enough, you get abnormal growth. And it could be of something external, which is infection, or abnormal growth of your own cells, which would be cancer. If you have too much reaction of your immune system to your own cells, you get an autoimmune disease. And in fact, a lot of times, remember that IL-6, overproduction of IL-6 in COVID is causing all those problems. That's a case of inflammation, where you have too much interaction. Your immune system is reacting too well to the outside world and damaging your own body. And here's another way to look at it. If you want to think of it as the yin-yang balance, or literally as a seesaw balance, here they have different kinds of, these are just different kinds of responses that you can have for the signaling molecules that are the interleukins. For in fact, you see IL-4 here and IL-12. You don't see IL-6 because there's a lot of them. But the idea is that you have a balance between the different things, and a lot of times you'll have weird connections between them, like the system that responds to intracellular viruses is sort of opposite from the system that responds to extracellular infections. And then you have helminths. Anybody know what helminths are? What's another word for a helminth? Worms. So parasites that infect your body. You want to be, but then you also have commensal bacteria. You don't want to kill all the bacteria in your body because you've got good bacteria in your gut. So it's all a balance, and you can talk about the balanced health in this case is a balance of all these signaling molecules. When autoimmunity happens, you have too much of the response of the type 3 response, too much of the IL-23, or maybe too little of the IL-17 that blocks the response. You know, it's things like that. So it's all very complex. But the cool thing about this, all these little yellow boxes are proteins. And they can be made and manipulated with protein chemistry. So this is the big thing that I want you to take away. This is related to your question. The fact that you have different speeds to different responses. If you already have antibodies around, or if your innate immune cells are activated, then they react right away, but they don't learn. They don't. It takes time to learn, and so you have 7 to 10 days is what it takes for your adaptive T cells to learn about something new and to make a molecule that will shield you against a new infection that it's never seen before. And in fact, if you know for all your childhood vaccinations, you don't have to keep getting those, right? Because you have memory cells that literally will last as long as you. And they will remember. They get a little bit foggy, just like we all do as you get older. But for the most part, you can have an immune response remembered for your whole life in some cases. So the virus goes up, you stop it going up, and then the virus goes down, especially when you have the adaptive response for a pretty serious infection. Question?
Was the concern with COVID immunity because of breaking out which it did that would cause it to not be
Well, the first problem is that you had no pre-existing antibodies, or that you had hardly any. I actually think you probably had some pre-existing antibodies, because it was somewhat similar to other coronaviruses, but it was essentially novel. So the first worry was that step one basically didn't have anything pre-existing already. The other worry is the worries about the papers that I showed at the beginning of this talk were more of the worries about step 3. Will this coronavirus not be remembered? And we found out that it will be remembered, but it took us months to find out that literally, yes, it will be remembered months later. You have to wait.
But
Right.
was that worried because of the virus or
Right.
because of the memory cells?
Well, basically what you have is because the virus was new. But the question is, how new was it? Because it was recognizably a coronavirus. And by new, we mean it was 90% similar to the original SARS, but the original SARS had not gone worldwide. It was 60% similar to other coronaviruses, which were worldwide. So when we say new, we mean really 40% new. But we didn't know, how new was that? Will it be like it's totally new? Will it be like when you have a population that's never been exposed to smallpox before? It devastates them.
There's such a massive difference in genetic, like 60%
Mhm.
is between E and E.
Right. Right. So you can look at it a number of ways. On the other hand, there are you can look at the different branches of the immune system, and it's reasonable to think that especially the T cell response should have some pre-existing response. It's much more reasonable to think of it a pre-existing T cell response, by the way, than an antibody response. And that is one of the main points I want you to get from today's lecture. Why can I say that confidently? Yeah.
I remember the vaccine first came out, people were saying that if you had COVID, you had a more adverse side effect to the vaccine.
Hmm.
Is that because of the pre-existing reactions?
Yeah, and when it comes to side effects, it's weird because we're talking about things that really are on the edge of what the immune system's supposed to do. And so there's a lot more variability when it comes to side effects as opposed to a more defined endpoint like hospital, going into the hospital from it or not. So I believe that it was always pretty clear that the best thing was to combine both natural immunity and natural infection and vaccine protection, that they would add up. And one of the questions was, will they add up to be, like 1 + 1? Will they add up to be 1 + 1 = 3? Or will they add up to be 1 + 1 = 1.5? Will they be synergistic or not? The good news is it seems like they're probably synergistic. And that's some of the details that we have. But we didn't know, right? And that was one of the big questions. And so, some people are still saying, by the way, I still see this stuff. This is why I should stay off of Twitter, because it bugs me that people are still saying this. But they're saying it attacks your immune system. It depletes your T cells. You don't really get infection to it. You don't really get protection against it. And I'm like, it's so infectious that you don't really get protection against infection necessarily, like a year later. But you get protection against hospitalization. And so you got to be nuanced about it. And I see all these people that are like, I literally saw a tweet that said, this is probably going to be the end of society. And when you see a tweet that says that, you probably should shut it off. But it was such bad reasoning that it still bugs me now. Because I'm like, yeah, it's bad and it's terrible and we need to do something about it. But I really don't think we're talking about the end of society here. And you've got other things that are going on here. So anyways, that's why I got to say, it does not appear to, that those statements that it attacks your T cells, it depletes your immune system, it doesn't look that much different than what a novel coronavirus should look like. It's a bad disease, but let's not make it worse than it is. So, this is the major thing that we talked about many of these things before. We talked about macrophages and B lymphocytes, B cells. T cells are the ones we haven't talked about, and there's two basic branches of them that we are going to talk about. There's cytotoxic, otherwise known as killer T cells, and then there's the helper T cells, which the killer T cells are able to directly destroy their target cell. But the helper T cells, they actually will interact with all the other parts of the immune system. In fact, to make antibodies, you have to be able to have helper T cells helping to make antibodies. So these are what we're going to talk about, 3A and 3B. Now, if you go into immunology, you can talk about where all these come from. And the main thing is these all come from the bone marrow, and these all come from the same kind of stem cell that gives rise to erythroblasts. What's the common name for erythroblast? Yeah, red blood cells. You see, it's basically, that's what it is. So, and even platelets come from this. All of these things like macrophages come from this. But way over here, you can have white blood cells that come from the same source as red blood cells. And so you have B cells and T cells that eventually, when they are activated, they become like the grown-up, productive signaling members of their thing. There's our macrophages right here. This, by the way, is my favorite immune text. And if you want to know my recommendation for it, I just love how it does it because these are, number one, realistic pictures of them, but they're also stylized enough that you can remember. So I've got some immunology teaching materials if you ever have a question about that. This is what they actually look like. Here's when it's resting, when it's activated at first, and then it grows up into an effector cell. This is the cell that makes antibodies. This is the cell that's a T cell. You can see at this perspective, you can't really tell the difference between them, right? But you know that this has B cell receptors on the surface. This one has T cell receptors on the surface. And they have very different jobs. So the main thing I want to close out this part with is that you have metabolic cycles that are connected to the immune signaling cycles. And when you have these, so that's why it's important to have Biochem 2 in the background. You don't need to remember specific things, but hopefully you remember the whole concept of the metabolic cycle. Your sugar cycles, if they have a problem, you can have a problem with your immune balance. And that's why you have complications from things like diabetes that have so many surprising results. For example, this is from the flu. If somebody with diabetes has higher blood glucose, and that means they have more of this UDP N-acetyl glucosamine sugar, which is used for building things, if you remember from Biochem 2. But you end up with too much sugar being built, too much sugar on this immune response factor 5, which means you have too many cytokines made. Therefore, a person with diabetes can have inflammation problems. And they're connected actually pretty directly by these two things overlapping. This is the exact article about it. And just so you had to know all these things, not because somebody's going to walk up to you someday and quiz you about the pentose phosphate pathway, although maybe you'll be on Jeopardy, and maybe they'll have that in there, and I'm not sure I could pass that one. But at least I know in general what all these things are for, and I know if I need to drill down on it, I can open up my book and drill down on it. The main thing is these are inside the cell. They have to do with energy, but they are connected to the signaling. When a cell is activated, it's going to grow. It might divide. It's going to do more of these cycles. And that will have chemical consequences in both metabolism and in the immune system. In fact, there's a whole field called immunometabolism that deals with these kind of questions, okay? So here's just an example of immunometabolism. When you have, here you have the three kinds of T cells. You know, there's the old riddle about what walks with 4 legs in the morning, 2 legs at noonday, and 3 legs in the evening. And it's supposed to be like a baby, a man, and then the old man. It's like the Sphinx. It was like one of those Greek stories. This is like the baby. This is like the human, the adult. And this is like the old adult who remembers things. If you have the baby cells, they get activated by an antigen, and they become effector T cells. They're doing their job. They switch from catabolic metabolism to anabolic metabolism. Instead of burning sugar, they are building. So it's like they go on the job for construction, and they make more of themselves. Then after the problem goes away, some of them survive and switch to being memory T cells. And the memory T cells switch back to catabolism. So the different immune functions go with different kinds of metabolism. And then you can get down to the molecular biology of what are the genes that help with this switch inside the cell. Here's a great figure that just shows, just want you to see, this is a lot of the stuff that you learned in Biochem 2. You have glucose coming in, Krebs cycle, citric acid cycle, whatever you called it. And then you even have chapter 19, oxidative phosphorylation going on here. And it's showing, you even have glutamine down here. This is the same structure they have in all of these, but they're going to show you, for example, which cell is this? Here's a cell that is doing aerobic glycolysis. So it's putting out glucose as lactate that's secreted. And it's turning on these pathways, but the dotted ones are not turned on. This is doing catabolism, but it's not really doing anabolism. It's not really building up any of the green things. And so you can have this aerobic glycolysis will support building anabolism. And so this is the one that I really want to show you. This is where we can see it. The blue is aerobic, and the citric acid, the blue is aerobic going down, and the red is cells that are tearing down. They're in an anaerobic mode. And you can see that you have glucose coming in, lactate coming out, in all three of the red ones. These are the different types of immune cells that do the anaerobic kind of metabolism. Whereas the cells that are building up are these different ones. Naive ones are building up here. Memory T cells are building up here. And you have they're sort of maintaining themselves. They're working with oxygen. When you have the cells that need a lot of energy, they will start to run anaerobically. And so the different types of immune cells have different types of steps that are activated. And you can classify them according to this. And it's helpful for you just to have in your head, oh, okay, this is what's going on. And in general, you have lactate being produced by all the red cells. But you have slightly different, like this one is producing nucleotides. This one is producing more lipids. They're all producing amino acids. So it's the question of what is going on inside the cell. And it's really complex. Even this is really too simple. When you look at the figure, there's a lot more going on. But needless to say, I just want you to know that this field exists. And it does tie these things together. You can have tons of different T cells. Look down here. You can have the different types will have different types of responses. They'll upregulate different types of cycles inside the cell. These ones on the right will upregulate oxidative phosphorylation. These ones on the left upregulate glycolysis. And you see that there's 6 different kinds of T cells that they're classifying right here. So there's a lot of different things, and I just saw this on Twitter, which is scientists complaining about nomenclature. So if you thought nomenclature was annoying when you were first got into organic, then it continues. And immune systems, yeah, like there are some people who are like, oh, I've named a new kind of cell. We call it the CD8-GINFKIR, and nobody can, even these scientists, these are research scientists running labs, and they're like, no one can remember that. Biologists would rather use each other's toothbrushes than each other's nomenclature. And that goes for immunologists. Everyone wants to have their own nomenclature. So all that is to say, we're going to stay at the level of there's 2 kinds of cells in the world, 2 branches of immune system. We're definitely simplifying. But it's also a very messy sort of ecosystem. It's not always clear which category cells go into. They maybe even change over time. But they're all important. And this idea that the immune system relies on a balance between self and non-self, where you don't attack yourself too much, but you also attack the bad things that are trying to attack you, unless they're not bad, and so you don't attack them. You see how it gets kind of confusing. And so it's this whole kind of balance where you have a whole bunch of different roles that are interacting. If one part gets out of whack, then the whole system suffers. And that's actually a very similar idea to what Paul is talking about in 1 Corinthians 12. The idea that the members have been organized, and it's proper to say God has organized the members. We're not imagining a ghostly hand moving everything to its right place. It's a very different kind of organization. And yet the ultimate goal is that each one is placed in the right place. There are many members, but one body. And it's not a machine. It's a biochemical assembly that's very messy, and it's not like either 0 or 100%. Sometimes it's 60%, 40%. And in fact, for the question with the coronavirus, we have something that's 60% old, 40% new. Well, how bad is that? And nobody knew at first, right? We still don't know for sure, but we know that at least we can fight this off. So it's a messy system of contradictory impulses. I have to drop one name right here. Samuel Taylor Coleridge actually had an idea called polar logic that says that the whole world is like this. And I would love for somebody to kind of put this together someday, but not me, not today. The idea is that the system is messier, but it's also less prone to complete collapse. Because if you don't have to be 0% or 100%, if you can live with being 90%, 80% right, your body can continue. And if that's continuance is the point, not perfection. And this is how evolution works at the big scale. And at the cellular scale, it's how it's how evolution even makes antibodies, if you remember how the clonal selection works. So that is the part that